Welcome to the fascinating world of Parazoa. Today, we are going to explore the simplest multicellular animals on our planet. We will discover how creatures without brains, hearts, or muscles manage to survive and thrive in the ocean. By the end of this lecture, you will understand not just their biology, but the elegant physics that keeps them alive. Let us dive in. Now that we know what topic we are covering, let us start with a simple analogy to understand what a parazoan actually is. Imagine you are trying to build a house, but instead of using cement to bind your bricks together into solid walls and distinct rooms, you just stack the bricks loosely. Each brick does its own specific job, but they never truly fuse into a single complex structure. In the animal kingdom, this is exactly how the simplest multicellular creatures operate. They are a cooperative community of individual cells living together in one body, making them the perfect bridge between single-celled organisms and complex animals. Building on that brick analogy, let us look at the formal biological definition. In biological classification, Parazoa is a subkingdom of animals that literally translates to beside the animals. A parazoan is defined as a multicellular, heterotrophic organism that exhibits a cellular level of organization. This is a crucial point. It means the organism completely lacks true tissues and organs. While their cells are specialized to perform different tasks, such as catching food, building the skeleton, or reproducing, these cells are never tightly bound into coordinated tissue layers like we see in other animals. Since these creatures lack true tissues, you might wonder what they actually look like in the wild. The most famous and practically the only surviving members of this group belong to the phylum Porifera, which we commonly know as sponges. Because they lack a nervous system, muscles, and a digestive tract, they cannot hunt or move like other animals. Instead, they rely entirely on a constant flow of water through their porous bodies. This continuous water current is their lifeline, bringing in fresh oxygen and food while simultaneously washing away their waste. Understanding how sponges feed brings us to an important question. Why should we care about a creature that just sits on the ocean floor? First, parazoans represent a crucial evolutionary milestone in the history of life on Earth. They show us exactly how single-celled organisms first began to collaborate and form multicellular bodies millions of years ago. Beyond history, they are vital to modern ecosystems. Sponges act as the ultimate water purifiers of the ocean. By filtering thousands of liters of water daily, they keep marine environments clean, clear, and perfectly balanced. Aside from cleaning the ocean, their stationary lifestyle has led to another incredible benefit for humanity. Because sponges cannot run away from predators, they have had to evolve entirely different survival strategies. Over millions of years, they have evolved to produce incredibly powerful chemical defenses to deter anything trying to eat them. Today, scientists are actively studying these unique, potent sponge chemicals. They are using them to develop new, life-saving pharmaceutical drugs, including advanced antibiotics and breakthrough cancer treatments. So these simple creatures are actually saving human lives. Now that we appreciate their importance, let us look at the physics of how they actually pump all this water without a heart. Sponges draw water in through thousands of tiny microscopic pores called ostia and expel it through a single large opening called the osculum. We model this using the principle of continuity from fluid dynamics. The formula is A in times V in equals A out times V out. A in is the total combined cross-sectional area of all the tiny entry pores, and V in is the velocity of the water entering them. On the right, A out is the area of the single large exit hole, and V out is the exit velocity. Conceptually, this means the volume of water entering must exactly equal the volume leaving every second. If the exit area decreases, the exit velocity must increase to push the same amount of water out. Before we move on, let us see this formula in action with a concrete example. A marine biologist is studying a sea sponge. We are given that the total combined area of the entry pores is 50 square centimeters, and water enters at a slow velocity of 0.2 centimeters per second. The exit hole has an area of just 2 square centimeters. We need to find the exit velocity. We choose the continuity equation because it perfectly models flow rate. We substitute our given values, multiplying 50 by 0.2, which gives us a total incoming flow rate of 10 cubic centimeters per second. Dividing this by the exit area of 2, we find our final answer. The water exits at 5 centimeters per second. Physically, this means the water shoots out 25 times faster than it entered, blasting waste far away so the sponge does not breathe it back in. Now that we have used the continuity equation to find velocity, let us understand how to calculate the actual number of pores a sponge needs. The formula for the total number of pores, N, is simply the total required input area, A in, divided by the area of a single microscopic pore, A pore. Conceptually, this comes from the fact that the total area is just the sum of all the tiny individual pore areas. If the size of a single pore decreases, the sponge will physically need a much larger number of pores to maintain the exact same water flow. We first find A in using our continuity equation, and then apply this simple division to reveal the massive scale of cellular cooperation. Let us apply this to a second example to see how many pores a sponge actually has. We are given a sponge that needs to expel water at 8 centimeters per second through a 3 square centimeter exit. The water enters at 0.1 centimeters per second. Before we can find the number of pores, we must find the total required input area. We rearrange our continuity equation to solve for A in. We do this because the laws of physics dictate exactly how much surface area is needed to support this exit flow. Substituting our numbers, we multiply 3 by 8 to get 24 and divide by 0.1. Our final answer for this step is 240 square centimeters. This is the total surface area required to keep the sponge alive. Following up on that calculation, we can now find the exact number of individual pores. We know the total required input area is 240 square centimeters. We are given that each individual tiny pore has an area of just 0.002 square centimeters. We choose our pore count formula, dividing the total area by the area of a single pore. Substituting the values, we divide 240 by 0.002. The final answer is an astounding 120,000 individual pores. Physically, this massive number perfectly illustrates the cellular level of organization in Parazoa. The sponge is relying on 120,000 tiny, uncoordinated openings working together simply to sustain the whole organism. Now that we have calculated the physics, let us visualize exactly what this looks like inside the animal. On screen is a cross-sectional diagram of a sea sponge. Notice the blue arrows on the sides. These represent the surrounding ocean water being drawn in through the thousands of tiny ostia we just calculated. The water enters the large central cavity, labeled as the spongocoel. Look at the walls of the sponge, labeled cellular layers. You can see they are made of individual cells, not solid muscle. Finally, follow the large red arrow pointing up and out. This is the osculum, where the water shoots out at high velocity, carrying away waste. This diagram beautifully summarizes how their anatomy perfectly matches the fluid dynamics we just learned. We have covered a lot of ground today, so let us review the key takeaways. First, remember that Parazoa, primarily the sponges, are the simplest grade of multicellular animals because they completely lack true tissues and organs. Second, they operate entirely on a cellular level of organization, acting like a cooperative community of cells. Third, because they cannot move, they rely entirely on the physical principles of fluid dynamics to filter massive amounts of water to breathe and eat. You have now mastered both the biology and the physics of these fascinating creatures, gaining a living window into the early evolution of all animal life on Earth. Great job!